Right, does anyone know anyone else who's coming? No? This is the audience then. So, hi, my name's Mark Pallen. I work at the University of Warwick, and I'm going to give a talk on Waking the Dead, Ancient DNA from Humans and Microbes. Now, in terms of an overview, first of all I have to apologise if this is a bit technical, because I basically taken material that I use in my lectures to students and just repurposed it. I have made some changes to try and make it a bit more accessible, but there's only so far I can go in the time available. And so what I'm going to talk about, a little bit about DNA, a little bit about ancient hominins, which is not my work, but I think is very, very exciting, and then a little bit about pathogens from the past, which is my own research. So starting with DNA, here's well, perhaps slightly overblown, eloquent account of what DNA is all about. Here he's saying it's raining DNA outside. He's talking about a willow tree pumping out its seeds. And one of the counterintuitive things is that all living organisms that are made of cells, every cell has DNA in it. So you're shedding DNA all around you. When you die, you shed DNA. Trees like this are shedding DNA. And that DNA is carrying the instructions to make new cells, to make new tissues, to make new bodies. And as he says here, it's raining DNA. It's not a metaphor, it's the plain truth when you're looking at this kind of thing. So DNA is what we call a polymer, a long polymer, and it's got a sequence of bases which are kind of like letters of the alphabet. In fact, we use these A, T, C's and G's to stand for the bases of DNA. And we can work out what the sequence of bases is by a technique known as sequencing. And one of the properties of DNA is that the sequence of DNA can change through what we call mutations. So in a sense, these are like spelling mistakes. So you've got a DNA sequence there at the top, and then you get a spelling mistake there which turns one of the T's in that sequence into an A. And if we line up the sequences, what we call alignment of sequences, we can identify these mutations and we can use them as a way of looking back into the past. And this approach actually dates back 50 years or so. The first conception that you might be able to use the information in DNA to look back into the past and use DNA as a document of evolutionary history goes back to these guys, Zucker, Cantle and Pauling, in 1965. Now what they suggested was that if you take a load of sequences from different organisms, so here we've got humans, chimps, gorillas, orangutans, and you line up a particular stretch of DNA from those organisms, the same stretch, most of the time it will be the same in closely related organisms like this, but every now and then there will be a difference, and you can tot up all these differences and you can use them to draw a family tree showing the relationships between those organisms. And so this approach using DNA gives us a completely different handle on evolution and reconstruction of evolution than what people used to do and what people still do, which is to look at fossils and look at the shape of things, the so-called morphology and so forth. It provides an independent molecular approach. Now, one interesting development in the last few decades has been the study of ancient DNA. So you can get DNA from historical archaeological material. So as I say, when you die, your DNA is spread into the environment, but it stays in your bones. Your bones turn into fossils, then maybe some DNA survives. And this gives us great opportunities to actually have a window onto the past and identify and characterize ancient sequences. And it gives us direct evidence of evolution at the molecular level. So when I was showing you a family tree drawn by DNA a moment ago, that was looking at living organisms, and you can track back from the sequences in living organisms. But it's so much more exciting if you can get direct evidence from material, historical or ancient material at that molecular level. And the thing is, because all living organisms have DNA, this is universally applicable. So you could use this kind of approach to analyse plants, animals, bacteria from the past. Now, there are many challenges looking at ancient DNA. As the DNA is liberated from cells, it gets degraded, and over time it gets broken down into steadily smaller fragments. It's a bit like taking a book that moulders away until it's just a few words left from the whole book. There is also the risk that as the DNA is damaged over time, you get changes in the sequence that you're trying to read from it. That's one issue. Another issue is that the amount of DNA that's often present in this old material is very, very small, and there's a great potential for contamination with modern DNA that's all around us in the environment anyway. And so it requires very careful lab work, and about 25 years ago there was this dinosaur DNA fiasco where someone said they found some dinosaur DNA, but it turned out it was just their own DNA that they were analysing. It was just a particular strange sequence in their own DNA. So we have to be very careful when doing this kind of work. Now that's a preamble about DNA. Now let's take a trip to the Neanderthal Valley. So this is a river valley near the German city of Düsseldorf, and it was named after a 17th century German pastor Joachim Neumann. Don't quite know why. It must have been a fashion at that time for the classical world that they translated his name Neumann, which just means Newman, into Greek, Neander, which means Newman in Greek. And Tal is the German word for valley, and the spelling changed in 1901. And it's a bit annoying because when you look at the scientific literature, some people spell it with the H, and some people don't. And it's actually become almost a British-American thing. So the British journals, like Nature, use the H. The American journals, like Science, don't. And it's annoying to see both of those when you're trying to do searches of literature. Now, the Neander Valley used to look a bit like this, a bit like Cheddar Gorge, very steep sides of this limestone gorge. And there are numerous caves and rock shelters along there. And as part of the industrialisation of Germany, they started quarrying the limestone and removing these valley walls and the caves within them. And as part of that process, they stumbled into a cave that was called the Kleine Feldhofgrothe and found a skull cap and some other bones. And these were initially thought to be a cave bear, but then they were shown to a local teacher and natural historian who said, no, these are human. They're human, but not quite as we know it. And they were written up in 1857 as a scientific report. And this is just the right time, because around that time Darwin was just about to put forward his Origin of Species and talk about evolution. This is the site today. I went there a few months ago, and they've actually marked with those poles there where the skeletons, they found very recently, about 10, 15 years ago, a whole load more Neanderthal bones. What it probably happened is that over here there's still this little bit of escarpment here, and all this was mined away, the limestone quarried away, and probably what was up here all fell down here, and that's why they found more recently Neanderthal bones there. But it's quite a different landscape now than it would have been when the original discovery was made, because there's such a flat area now around the sides of the river valley. Now, Neanderthals, or Neanderthalers as we call them, or Neanderthal men, were different from modern humans in terms of their anatomy. They are much more robust. They have this pronounced brow ridge, this projecting mid-face. Some of the features are annotated on this skull here. And here at the museum at the Neanderthal Valley, at the original, next to the original site, they've reconstructed what a Neanderthal would look like, but they put an interesting little twist. They put him in a suit, just to highlight the fact that they're not that much different from us. And there are plenty of bankers out there who've put Neanderthal in their attitudes to risk and morality and so forth. So yeah, maybe dressing him up like a banker is not a bad thing. Now, obviously, if you want to make a comparison, you have to take an anatomically modern human and put them side by side with Neanderthal, and that's what you get when you do that. And you can see there are some similarities and there are some differences as well. Now, one of the things that became clear over the last 20 or so years is that modern humans like us actually originated in Africa, and then we spread out over the rest of the world. And we didn't leave Africa until relatively recently. The exact date is still a matter of dispute, but on this figure here, about 72,000 years ago, they say that humans spread out of Africa, spread into Europe, spread into Asia, and from Asia went into Australasia, and very much later then peopled the New World as well. Now, one of the interesting facets of this is that as modern humans came out of Africa, and this slide is, this figure here is a little contentious really, because it's unclear quite what route they took out of Africa. They may have gone out through the bottom here of the Arabian Peninsula instead. But as they came out, they would have moved into the area that was occupied by the Neanderthals. So the Neanderthals were living in Europe and West Asia at that time. Now that raises an interesting question. What happened to the Neanderthals? So the last Neanderthal skeletons that we can find, I think they're about 40,000 years old, clearly Neanderthal. So what happened? So Neanderthals here are kind of branch on the human family tree that come off here and then die out. Modern humans, we evolve in Africa, we diverge in Africa, and then a group comes out and populates Eurasia. But the question is, when we came out of Eurasia, was there any interbreeding with the Neanderthals? Or we wiped them out and that was the end of it? Or they died for some other reason and that was the end of it? So this is an interesting question, and it's a question that we can address using DNA. And the first efforts to try and address this question looked at a source of DNA known as mitochondrial DNA. So each of your cells has got many copies of this mitochondrial DNA. It's what we call an organelle. It lives inside your cell, and it's slightly unusual because it's got its own little bit of DNA in there, separate from all the rest of the DNA. The stuff you learn about in biology at school, where you have all those chromosomes, the mitochondrion is slightly separate from that because it's got its own small chromosome. And the reason this is favoured by people looking at ancient DNA is that there are lots and lots of copies of this per cell, and it evolves very quickly. It's quite a small genome, and it also gives you quite clean answers because it's passed on down the maternal line. So a man will get his mitochondria from his mother, daughter will get her mitochondria from her mother, and the daughter will pass the mitochondrial sequences on to her offspring. Now, the first evidence on mitochondrial DNA was gathered in the 1990s through around to the turn of the millennium, just after the turn of the millennium, and it gave us what appeared to be a very clear signal. So if you draw a family tree from mitochondrial DNA extracted from Neanderthals, the Neanderthals sit on this family tree well outside all the variation that we see in modern humans. So it looks as if Neanderthals are, if you like, a separate species. They're quite different from us. And suggests that there was no interbreeding between us and Neanderthals. And these colours here, this just shows you the accumulated evidence of Neanderthal sequences compared to the modern human sequence at the top there. So it's just colour-coded. And the reason that I put that up there is to show that one of the questions that was raised, the query that was raised, well, you get this sequence out of Neanderthal bones, but maybe it's just damaged, and it looks different because it's damaged. But in fact, when you look at multiple samples, you get the same kind of picture, suggesting that really they are distinct from modern humans. And that was the way it was. That's the way what everyone believed until about 7 years ago. And then things changed. Because then we actually got sequences from the rest of the Neanderthal genome. So those 23 chromosomes and stuff you learn about in biology, we started to get that kind of information, which is much more comprehensive, many more data points in there. And this effort really was down to the work of this one man here, Svante Pääbo, who's been a true pioneer in this field, and over the last couple of decades, maybe 3 decades, has really been pushing forward the boundaries of doing this ancient DNA research. So we were all very excited when we saw this Neanderthal genome, but the question what you need to address is, are Neanderthals more closely related to some modern humans than others? So if they'd bred with modern humans at some stage, then some modern humans should be more closely related to them than the ones that didn't breed. And so when they got this first draft of the genome of the Neanderthals, they did some comparisons to a couple of European Americans. One of them was Craig Venter, a couple of East Asians, and 4 West Africans. And what they found was that Neanderthal genomes showed a much greater similarity to the genomes from the non-Africans than they did from to the Africans. So this was a little bit surprising, because everyone had kind of painted the Neanderthals as a European phenomenon, and you kind of thought, well, if there were going to be any signal here, it would be in Europeans, but you wouldn't expect to see it in others from outside Africa. But that wasn't what they found. What they found was, when they did these comparisons with different ethnic groups, they showed that all people who are from outside of Africa, so people like us, but Australian Aborigines, people from Papua New Guinea, Chinese people, they all showed some Neanderthal ancestry. And in the paper, I think, I'm not sure it was in the paper or in the press release, they made the point that Craig Venter, who at the time was in the news a lot because he'd been involved in the Human Genome Project, there were segments of his genome that were more Neanderthal than they were African. And the figure was around 1 to 4% of non-African genomes are Neanderthal. Now, things have moved on in the last few years, so now you can actually go to a company called 23andMe, and you can spit in a tube, and they will extract your DNA, and they will genotype it for you, and they've done this for me, and I am 2.8% Neanderthal, which is pretty much the same, the average, for the average 23andMe user, the average European is about 2.7, 2.8%. I think my daughter is 2.7%, which means my wife must be even less Neanderthal than she is. So that's the kind of gradation of Neanderthalness in our family. What this means is that we have this divergence between Neanderthals and modern humans. Then within the modern human branch, we have various divergences. So the earliest branch is this branch called the San, who's a bushman, live mainly in southern and southwest Africa now. And then branches coming out from at this point here where humans left Africa. And sometime around the time we left Africa, maybe before, maybe after, Neanderthals interbred with modern humans and left some of their genes living on in we non-Africans. And this area, this research area, has actually been very exciting over the last few years. Now people are actually going back and looking at modern human genomes and resurrecting the Neanderthal lineages within them. So this is a way of getting the DNA, the sequences of these Neanderthals, which are long extinct, from modern humans without using ancient DNA approaches. You're using ancient DNA to work out what looks like a Neanderthal in the first place, but you can then go back in and do more studies as they've done here. Now, when I first it was a few years ago when I gave this lecture, David Tennant was playing Hamlet, and the first year we got the Neanderthal genome, the second year we got Neanderthal. It was amazing. But then it got even more amazing because there was something turned up that nobody had expected. And this is one of the exciting things about ancient DNA research, is that it sometimes turns up things that you just didn't expect to be there. So a little bit of a finger bone was found in a cave in Siberia. This size thing here. And they extracted DNA from it. And what they found was a genome of an unknown kind of human that had never been described before. When they drew a family tree based on the DNA, all this lot here is modern humans. In the blue, it's not a good projector, it's focus. This little blue bit are the Neanderthal sequences. And over here is the sequence from that little finger bone from this cave in Siberia, so-called Denisova cave. And the name for these individuals was called the Denisovans. And it turned out that they were an outgroup. They were clearly more distantly related to us than Neanderthals were. In fact, more recently, it appears that they are more closely related to Neanderthals than they are to modern humans. And we keep getting more and more data from these individuals as time goes on. Now, the weird thing, though, was that not only had they branched off a little bit earlier than Neanderthals, but they had left some residue of their genome in modern humans, but only in one particular branch. So in the initial studies in the Papua New Guinea branch, so-called Melanesians, turns out also they're found in Aborigines. And this is a bit strange because the one piece of DNA that started all this came out of this bone found in Siberia, and Siberia is a long way from Papua New Guinea, and it's a long way from Australia. But somehow when modern humans marched out of Africa and through Asia, they came across these people, the Denisovans, and the Denisovans interbred with them and gave some DNA. And coming back to the legacy of this, you could say, well, it's just, so what? Does it really matter? Doesn't actually change the world, this interbreeding, has it? Well, it has, because modern Europeans and Asians actually have this residue of Neanderthal DNA, and some aspects of that DNA are controlling quite important characteristics. So things like the colour of your skin. The fact that I've got this stupid skin that when I go out in the sun it goes all red and blotchy and burns, and that is probably the legacy of Neanderthals. And there's a paper that just came out recently where they said that actually quite a lot of modern illnesses, if we look for the genetic basis of them, and then we look at where that genetic basis fits in terms of Neanderthals versus kind of African modern humans, it looks more Neanderthal. And the same thing's been found to be true for Denisovans as well, so that they actually do contribute to our biology in important ways. So it's interesting we've got this residue of this. One of the interesting things about this field, though, is every year, in fact every few months, something new and exciting pops up. So this was, I think it was last year this turned up. Here they actually found an early modern human who had a Neanderthal ancestor really recently. So it was some modern human remains, but when they actually sequenced the DNA, they could say it was equivalent to great-great-grandfather, a great-great-great-grandfather or grandmother would have been a Neanderthal. So that's kind of really weird. So about 40,000 years ago, and these green splodges here, this is a map of all the chromosomes, these green splodges are the Neanderthal parts that are in that genome. And it's in the last year or two, it's got actually more complicated. So it seems that we're getting DNA coming into modern humans from Neanderthals. They've just recently realised that there was some flow of modern human DNA back into Neanderthals. So it wasn't a one-way exchange. And it seems that there was some branch of anatomically modern humans that donated some genetic material to Neanderthals, but only to the Neanderthals found in the eastern range. So the ones found in Siberia, the ones found in Western Europe and Central Europe didn't have this DNA. So that's kind of weird and interesting finding. And it seems that all sorts of branching off, and then interbreeding were going on. So there's the Denisovans, there's supposed to be this other shadow branch that we don't quite know where it's come from, that may have donated DNA. So things are looking very, starting to look more complicated. We started off with a simple story that humans left Africa and they didn't interbreed with Neanderthals. Then we said, okay, they did interbreed, but probably only did it once. Now it appears that there's all sorts of interbreeding going on multiple times. And just this week, we have seen some interesting results come from this place, Atapuerca, in northern Spain. And this is a series of caves and a trench that's been dug, and is the site of the earliest known human remains in Europe. And I had the privilege of going there a couple of weeks ago and having a look round. And one of the interesting things is there's this pit called the Pit of Bones, where there's this long pit here, and the bones are found at the bottom. And I think there are 28 individuals have been reconstructed from those bones. And a couple of years ago, they actually extracted some DNA and got a sequence from those bones, from one of the bones. And they then got a rather weird result, because the bones that were found, when paleoanthropologists look at them, they say they look like modern humans, but a kind of bit Neanderthal as well. So maybe, well, they look like the ancestor of Neanderthals and modern humans. They got a touch of both. And they were expecting them to come out looking a bit Neanderthal. But in fact, when they looked at the mitochondrial sequence, they found that they were closer to the Denisovans. And that's a weird thing, because the Denisovans, hang on, they're in Siberia, and the legacy in modern humans is out there in East Asia and Australasia. What on earth is a Denisovan sequence doing out there in Spain? And that was the way it was just a weird finding, and it still is a weird finding. And then on Monday of this week, a paper came out in Nature where they actually got some nuclear DNA from all the other chromosomes and stuff. They didn't get a whole genome's worth, they didn't get very much DNA. They got enough sequence to be able to say, actually, the main part of the genome is Neanderthal. So we've got this weird thing where we've got these humans from Spain who have a nuclear genome that looks a bit Neanderthal and a mitochondrial genome that looks Denisovan. And we still don't really understand what's gone on there and how that actually maps onto what we know about these archaic humans and their distribution. So that was all I was going to say about archaic humans, about Neanderthals and Denisovans. If you'll allow me to just continue for a few more minutes, I'll take you on to some of my own work. And the story for this begins in Central Europe, a bit further east than Neanderthal. Now we're in Hungary, on the banks of the Danube, in a town called Vác. In 1994, there was a crypt discovered as they were trying to do some renovation on this Catholic church, Dominican church, in the heart of the city. And what they discovered was this crypt had been sealed up for over 100 years, over 150 years in fact. It had been used for the burials of middle-class Catholics from 1731 to 1838. And strangely, when they opened the coffins, they found that many of these individuals had become naturally mummified. So instead of all their flesh rotting away and just leaving a skeleton, there was some of the soft tissue left behind. And we started getting interested in this about 3 or 4 years ago, and we focused initially on this lady called Teresia Hausmann, and she died on the 26th of December 1797 at the age of 28 years. And in fact, this here, you can't read it very clearly, this is the result, the record of her death in the register. So it was interesting to have humans that have got soft tissue, well dated, to work on. And a lot of work was done on these mummies in the years following their discovery, around the turn of the millennium. There's a guy called Mark Spiegelman, who's an Israeli, British, Australian archaeologist and surgeon who went in, and they did all sorts of investigations, did chest x-rays and other investigations. In her case, her chest x-ray looked okay, but she did appear very wasted, which is consistent with a diagnosis of TB. And it was found among these mummies that about half of them had some evidence that they had TB, tuberculosis. And in her case, a variety of analyses showed that she actually probably had quite a lot of TB DNA left in her tissues, particularly in her lung tissue. And so we thought, well, let's see if we can get any genome sequence out of that, if we can actually just sequence the DNA extracted directly and see what we found. And what we ended up doing was we ended up working on her, but we ended up working on in total 8 of these bodies, and we managed to actually reconstruct genomes from the organism that causes TB, Mycobacterium tuberculosis. And all of these genomes belong to a particular lineage known as the Euro-American lineage of TB, so not surprising in that we're in Europe. But one of the strange things we discovered was that in most of these bodies, we found more than one TB strain. Now, I'm medically qualified, I'm a medical microbiologist by training, and I've come across this, I thought, this is a bit weird, isn't it? I mean, normally when you get TB with just one strain. That was my assumption. I had to go and look at the literature, very carefully search the medical literature, and then we discovered that actually there is evidence out there in some of the papers to suggest that even today people can have more than one strain of TB. And it kind of, looking at this ancient DNA, made us realise that perhaps we were being a bit naive to think that people only had one strain at a time. Then what we did was, having these 200-year-old genomes, we could draw a big family tree of all of the TB genomes that are available, and try and work out when this particular lineage, this Euro-American lineage, first arose, where it was the most recent common ancestor, and it turns out it was about 400 AD, about the late Roman period. And that was a very interesting finding, but the mutation rate that we calculated to show how much change had to have been in the lineage for it to come from that date, it conflicts with some other evidence out there. Basically, it suggests that TB is actually really quite a young disease, and there are people out there saying, well, actually they've got some TB from Neolithic samples, and this kind of conflicts. And so we're currently working with some people in Israel, Mark Spiegelman and his colleagues, to see if we can get any sequences out of the Neolithic samples. Right, last thing I was going to tell you about, now we get on to skeletons. We've spoken about mummies. So we got involved with an Italian physical anthropologist, Raffaella Bianucci, who's shown there in the corner. And these are 2 of my postdocs who did the work. She basically gave us material from this skeleton 2568, which was discovered in a grave from this medieval village of Gerudo, which was found in northern Sardinia. And it had been buried in the second half of the 1300s and excavated in 1997. And when it was excavated, around the pelvis here, and this part here, there were these funny little nodules. And we don't know what those nodules are. They're about a centimetre across. But we were given some of them in our lab. And Jen McKay, my postdoc, extracted DNA from them. And we sequenced the DNA. And what we discovered was an organism called Brucella in the sequences from Brucella in that DNA, in that sample. And using the DNA sequences we've got from the sample, we could actually place this on a family tree of all the current Brucellas that are known. We could show that it's actually quite closely related to Brucella from Italy. Now, you may or may not, probably most of you haven't heard of Brucella. Brucellosis is an infection which has a long history, goes back. You can see certain changes in the skeleton that are diagnostic, and these have been seen in the skeletons from the beach in Herculaneum when the eruption of Vesuvius buried those individuals. And the organism that causes Brucella melitensis, is acquired from sheep and goats. And And this fits with the idea that there's been a long history of sheep and goat farming in Sardinia. But we don't quite know how this skeleton got infected, how the individual got infected. And there are two main routes, plausible routes here. One is that he was a farmer who came into direct contact with sheep and caught the brucella that way. Or it may be, and this is a well-accepted route of transmission today, he ate dairy produce and he got infected that way. But whichever way he got infected, this is an interesting insight into a disease in a medieval population. So I've now just about finished, 5 minutes for questions. Interestingly, ancient DNA studies have shed light on the flow of genes between what we call archaic hominins, Neanderthals, Denisovans, and modern humans. They've shown that, we're looking around this room, everyone in this room is a bit Neanderthal. 2.8% of you is Neanderthal. We've shown in my own group that we can use ancient DNA studies to provide insights into pathogens in the past. And the exciting thing is that it's just going to get more and more exciting because work is always ongoing in this field. Every few months there's something new comes up with some exciting new twist in the tail. And that's me finished. Just acknowledge the people who have helped and people who funded us. If you want to know more about this kind